appearance. If one aim is to find out genetic difference between the two individuals, okay, sequencing of DNA is the best method. Next, imagine try to compare two sets of 3 into 10 to the power of 6 base pairs, and DNA fingerprinting is a very quick way to compare DNA sequence in two individuals. DNA fingerprinting involves identifying differences in some specific regions. In a DNA sequence called repetitive DNA, because in these sequences a small stretch of DNA is repeated many times. These repetitive DNA sequences are separated from bulk genomic DNA as different peaks during density gradient centrifugation. The bulk DNA forms major peak. Other small peaks are referred to as a satellite DNA. Depending on base composition, adenine thymine ratio, guanine and cytosine ratio rich. Length of the segment, number of repetitive units. At the same time, satellite DNA is classified into many categories such as microsatellites and macrosatellites or mini satellites. It is mini satellite, etc. These sequences are normally do not code for any protein, but they form large portion of human genome. These sequences shows high degree of polymorphism and form the basis of DNA fingerprinting. Since DNA from every tissue such as blood, hair follicle, skin, bone, saliva, sperm, etc. For individual, they shows the degree of polymorphism. They become very useful in identification of forensic applications. We have a BSc Forensic Science. Further, polymorphism, if you carefully observe, inheritable from parents to children. DNA fingerprinting in the basis of paternity testing in case of disputes. As polymorphism in DNA sequence is the basis of genetic makeup or genetic mapping. If we take the human genome as well as DNA fingerprinting, it is essential that we understood that DNA polymorphism means, in simple terms, polymorphism, variation at genetic level, arises due to mutations. It is due to mutations. Okay. In case new mutations may arise in somatic cells or germ cells. The mutations occurs in germ cells can easily inheritable through the sexual reproduction. If germ cell mutation occurs, does not seriously impair individual's ability, but to have an offspring who can transmit the mutation, it can spread to other members of population. Allelic sequence variation at the alleles has traditionally been described as a DNA polymorphism. In simple terms, inheritable mutation is observed in the population at a high frequency. It is referred as a DNA polymorphism. The probability of such variation to be observed in non-coding DNA sequence. Mutations keep on accumulating generation after generation and forming one basis of variability, polymorphism. Understood, everyone? There is a variety of different types of polymorphism, ranging from single nucleotide changes to very large scale changes. Okay.